Modern bioengineering has moved past simple chemical treatments into the realm of synthetic molecular architectures, custom-built tools designed for high-affinity disease detection and forced cellular repair. The efficacy of these tools depends entirely on their structural backbones. Two primary examples are peptide nucleic acids, or PNAs, and phosphorodiamatidate morpholino oligomers, known as PMOs. Look at the structural mechanics of a PNA. Unlike standard nucleic acid, which has a negatively charged sugar phosphate backbone, a PNA uses an uncharged peptide-like structure. This allows it to bind to target strands with remarkably high affinity. This high binding affinity makes PNAs ideal for highly sensitive in vitro diagnostics, where they can accurately detect cancer mutations or viral genomes in a lab setting. PMOs utilize a different geometry, replacing the standard ribose sugar ring with a morpholine ring. Because this morpholine ring resists enzymatic degradation and maintains a superior safety profile, PMOs excel in vivo. They are current staples in muscular dystrophy therapeutics. The primary limitation of PNAs in a treatment context is their difficulty with in vivo cellular delivery, especially compared to the more resilient PMO structure. While both architectures target specific genetic sequences, their structural differences dictate their use cases. PNAs for precise laboratory detection, and PMOs for resilient therapeutics within the body. These molecules carry significant logistical barriers. Both PMOs and PNAs require more complex and expensive synthesis processes than standard oligonucleotides. There is also the intellectual property landscape to consider. Any diagnostic tool inspired by an existing PMO or PNA structure must legally cite the original patent as prior art. If a newly engineered tool actively utilizes the specific chemical claims of an existing patent, formal licensing and royalty agreements are legally mandatory. Because of the high hurdles in gene targeting, researchers often look to extracellular structural cell signaling. This approach is particularly relevant for spinal cord injury, which presents a uniquely hostile bioengineering challenge. Immediately following a spinal cord lesion, tissue undergoes rapid necrotic cell death and apoptotic cascades, eventually forming an inhibitory glial scar that physically blocks recovery. In an environment defined by physical barriers and rapid necrosis, standard gene regulation moves too slowly. This led to a molecular solution that bypasses gene targeting entirely, using cell membrane penetrating peptides paired with a bioactive IKVAV motif to physically anchor a regenerative signal to the lesion. This nanostructure physically embeds into a damaged lipid bilayer, anchoring the structure while leaving the bioactive IKVAV sequence exposed in the extracellular space. Anchored directly into the spinal lesion, the peptide provides a sustained regenerative signal derived from laminin. To evaluate the impact of this signal, we can look at the comparative morphological analysis of the spinal gray matter 28 days post-injury. This micrograph shows tissue from control subjects treated with mannitol or the peptide alone. The motor neurons are heavily atrophied and shrunken. There is a distinct lack of active cellular processes and a visible loss of structural integrity. Now look at subjects treated with the complete IKVAV-peptide. The gray matter contains massive active motor neurons surrounded by thick nerve fibers. They grow physically larger than healthy uninjured neurons. The pale nuclei and dark nucleoli indicate massive reactivation. This evidence reveals that the peptide does more than stop cellular death. It forces the surviving neurons into a hypermetabolic state. We can verify this activity by scaling down from cellular morphology to subcellular ultrastructure using electron microscopy. At this scale, the control neurons show clear internal degradation, dispersed nuclear chromatin, and loosely segmented rough endoplasmic reticulum. IKVAV-treated cells feature massive rough endoplasmic reticulation and high mitochondrial density. Highly folded nuclear membranes prove elevated transcriptional activity. The analysis also extends past the neurons to the surrounding glial cell environment within the gray matter. The distinction here is vital. While fibrous astrocytes form detrimental scars in the white matter, protoplasmic astrocytes in the gray matter are necessary for axon regeneration. Enumeration data reveals that IKVAV-peptide treatment drastically increased the density of these beneficial protoplasmic astrocytes surrounding the reactivated motor neurons. At the ultrastructural level, the peptide successfully coordinates a dual response: massive internal organelle expansion and the recruitment of regeneration-supporting glial cells outside of it. The final test of this therapy is its ability to translate subcellular reactivation in the central nervous system into macroscopic function in the rest of the body. Following a spinal cord injury, subjects typically experience severe skeletal muscle atrophy and the rapid infiltration of intramuscular fat due to the loss of nerve signals. Comparing histological cross-sections, panels C, D, and E show control treatments failed to prevent severe atrophy. Contrastingly, panel B shows thicker, preserved muscle bundles, mirroring healthy tissue in panel A. The lack of white space proves reduced intramuscular fat. Localized anchoring of the peptide in the spinal lesion cascades outward, proving that subcellular rescue directly preserves the macroarchitecture of peripheral muscle. The localized physical anchoring of these signaling motifs actively overrides the severe biological degradation that normally follows physical trauma. By anchoring the laminin-derived sequence directly into the damaged membrane, the tissue is forced past preservation and into a compensatory, hyperactive state. This study sits within the broader landscape of modern bioengineering that we established earlier. We can compare the physical mechanism of this cell-spanning peptide with the binding precision of PMOs and PNAs. Whether a tool is designed to bind to an RNA strand to alter gene expression or anchor to a cell wall to stimulate neurite growth, the underlying philosophy is the same. The results from the IKVAV-peptide demonstrate the power of engineered synthetic architectures, molecules custom-built to survive hostile tissue and mandate specific biological outcomes. This technical breakdown was brought to you by Synestec Systems. If you enjoyed this video, please like and subscribe. We welcome suggestions for future videos. Today's video was brought to you by Synestec Systems LLC, founded by Shane Persinger.